A very good morning to my dear students of class 8, and I am happy to welcome you all to today's zoology class. What are we going to learn today? Our today's topic is going to be about blood groups part 2. What is this blood groups part 2? Why part 2? Already we have completed the topic blood groups, right? So this part 2 video is made to clarify certain doubts that was raised in the Zoom session. Okay, so the portions in your textbook is covered about blood groups, but maybe out of curiosity or wanting to know more about blood groups, there were some doubts raised by your students. If you have not attended the Zoom session, you will not know. Because there were some doubts asked about the blood groups. I will first tell you what the doubts were, and then I will give the explanation part. I have promised them that I will make a video on that, so that is why a part 2 on this blood group topic. Okay, so let us get started. If you have not watched part 1 video about blood groups, please watch that video and come back to this video because you may not understand anything. But before entering into the topic in detail about the clarification of doubts, I will give you a brief recap of the previous part 1 blood group topic, and then I will tell what the doubts were and give the explanation. So, in the previous part, in part 1, I had explained about two terms, antigens and antibodies. What are antigens? On your red blood cells in every body, in every human being's blood, you have RBC cells, right? On the surface of the RBC cells, you have a protein which is called as antigen. See, these are all RBC cells. Whatever is in red circle, no? These are all RBC cells. And blood groups generally, they are of 4 types, either A blood group, B blood group, AB blood group or O blood group. Out of these 4 only, any one, each one of us will have, right? I told you there are antigens on the surface of RBC cells, no? So, A blood group person will have antigens like this. Say for example, some circles on their surface. B blood group person will have a diamond like structure. Actually there are no circles or diamonds inside your blood, but just to differentiate, it is represented as circles and diamonds. Okay, so say for example, B blood group persons have diamond shaped structures, that is the antigens on their surface. So round is A antigen, diamond is or square is B antigen. Okay. What about AB blood group people? AB blood group people will have both antigen A and antigen B, that is circles and diamonds. What about O blood group people? They will have no antigens. There are no antigens at all on the surface of the RBC cells of O blood group people. Okay, and also you have one more thing in your body, in the plasma of your body, which is called as antibodies. What is plasma? Take your blood, remove all the cells in the blood. You have white blood cells, red blood cells, no? If you remove all the cells in the blood, you will have a remaining white color liquid. Okay, that liquid is called plasma. So, in the plasma of every person, there will be something called as antibodies. Where are antigens? On the surface of the red blood cells. Where are antibodies? In the plasma. Okay, so every human being will have antibodies in the plasma. Just like how the type of antigens vary according to the blood group, the type of antibodies will also vary. Okay, say for example, the plasma of blood group A person will have like this structure. Okay, how you can interpret this structure? This structure can fit with this. See, if there is a diamond, if this is the shape of the antigen, see, antibodies shape is like this. It will be like a lock and key method. Okay, it will exactly fit. At the same time, if you see this antigen, it is of this shape, right? So this shape antigen can fit into this shape antibody. Whatever I draw down, no, this is antibody, because see, this is antibody A. So antigen A and antibody A are like lock and key. They will fit exactly together. Okay. But if they fit, no, if they are going to fit like this, the blood will clot. The blood will not be a liquid, it will clot and it will lead to death. Okay, never there should be clotting of blood in your body because it should remain a liquid, no. So if you see blood group A person, a person who has antigen A will always have antibody B only. Like if this is the shape of antigen A, he will have antibody B which will not match, this will not match at all because see a diamond cannot fit into a curve, right. So A blood group person, antigen A person will have antibody, these are all antibodies, okay, antibody B but never antibody A. At the same time B blood group person, a person with antigen B will have antibody A. It will be opposite because never B and B will be present together, A and A will not be present together. Okay, if it is present then the person's blood will clot off. So always person B, that is a blood group person B with antigen B will have antibody A. So A antigen will have B antibody in the plasma, B blood group person with B antigen will have A antibody in the plasma. What about AB blood group person? AB blood group person will have antigen A and antigen B, but if you see the plasma there will be no antibodies at all. Okay. Suppose, imagine if there is going to be any one antibody in their blood plasma. Since they have antigen A and antigen B, even if one antibody is present, it will go and bind with the structure of A or B. Suppose if there is A antigen, it will go and fix, suppose if there is A antibody, imagine it is not present, imagine if there is going to be A antibody, what will happen? It will go and bind here and it will result in blood clotting, the blood will clot, it will not be a liquid. Okay, suppose if there is B antibody, okay, this is the shape of B antibody, no, if there is B antibody, what will happen? It will go and fit into this antigen and again the blood will clot. But since there is no antibody at all, there is no blood clotting, blood remains a liquid. Okay. Here O blood group people will have both antibody A and antibody B, but there are no antigens for the antibodies to go and fit into the antigen. So again there is no blood clotting. Okay. These are all basics. Now I'll tell what the doubt was. The doubt was how to test a blood, whether there is a instrument or device to test a blood. How do we find the blood group of a person? This was one doubt. What was the first doubt? How in reality people's blood are tested. I think you must have seen some health care professionals coming to your house and pricking your hand with the needle during the time of dengue fever all, no? Dengue fever was common once, no? So many used to come and take your blood sample, no? They used to prick with the needle and check your blood all. So that is, that's all you know, I think, no? About blood testing. What do they do with your blood? How do they confirm that even when you go for blood checkup, if you want to test your blood group, if you go to a laboratory also, they prick with a needle and get 1 or 2 drops of blood, right? So how do they conclude what your blood type is? This was first doubt, okay? And the second doubt was, along with your blood group, I told you there is AB, B and O blood group, no? Along with your blood group, if you know your blood group, you will have either a positive or negative symbol, no? Do you know that? You have like B positive is my blood group. So B is my blood group and this positive also I have. What is that positive or negative? That was the second doubt. Okay. So this video will be about how to test your blood and what is this positive and negative. Okay. Before that, you should know a term called as agglutination. I had been repeatedly talking about blood clot, right? I was telling blood clot repeatedly. See, in blood clot, I don't know how clear this picture is. Actually, this is normal blood and these two are clotted. Okay. This is normal and these two are clotted. Blood should not clot, no. If you have clots, you will have dots like this in the blood. Okay, like this it will be. If it is a normal blood, it will be like a fluid. Never a blood should clot. If a blood clots, it is not acceptable. Your blood should flow in your body, no, it should never clot. And this process is called as agglutination. If blood is going to clot, it is called agglutination. Agglutination is blood clotting. Now I will teach you how blood is tested. For blood testing, what you have to do is, first, in your finger, any with needle or pin or any with sharp device, it is pricked, okay. Your fingertip is pricked, and what you are going to do is, a clean glass slide is taken, okay. A glass slide is taken and it is labeled as three labels. One should be A, one should be B, and the other should be positive or negative. Okay, this is the protocol how blood is tested actually. So, a clean glass slide is taken, A, B, positive, negative, like that 3 labels are given. Okay, now prick your hand with the needle and then place one drop of blood near A, place one drop of blood near B and place one drop of blood near AB. Okay. Then there are 3 bottles. Can you see? Blue, yellow and white color. What are they? What they will have? The first bottle will have antibody. See, all of them are antibodies. Okay. Your blood will have antigen and you are going to test against the antibodies. So, whatever is in the bottles are antibodies, not antigens. So, this blue bottle will have antibody A, okay, and this yellow bottle will have antibody B, and white will have antibody D, okay. Okay, what is this D then? I told you, no, certain people will have positive blood group and some will have negative blood group. The presence or absence of antigen D only will decide whether a person's blood group is positive or negative. What is this D? See, apart from this A and B antigens and antibodies, in your body there are also, not all will have, but a few will have D antigen also. Okay. So, if a person has D antigens, then the blood group report will be positive. What is this D actually? It is rhesus monkey antigen. It is a type of monkey. This type of monkey used to have this type of antigen. From that monkey only we got that name rhesus monkey. Okay. So, certain people will have that monkey's antigen, and if they have, the blood group is positive. Even my blood group is B positive. So, I too have the antigen from the monkey. Many of you, whoever has positive blood group, no, we are all we have inherited the monkey's antigen. Okay. So, that is why it is declared as positive or negative. Those who have antigen D in their blood, they are declared as positive blood group people. Okay. So, now what you are doing? I told you the 3 bottles will have antibody A, antibody B and antibody D. Okay. So, we have 3 drops of blood now labeled as A, B and positive or negative. So, take antibody antibody A and pour it to the first blood drop. Take antibody B and pour it to the second blood drop, and take antibody D and pour it to the third blood drop. So, after pouring that, what the people who test used to do is, with the match stick or a toothpick, the antibody what we have added to the blood should be mixed well, and make sure for every mixing a separate toothpick or the matchstick's backside should be used. Okay. Because the result will interpret, if you use the same stick, no, you will take on the antigen and antibody from one drop to the another. So, for every mixing separate sticks will be used usually. Okay. So, after mixing, see now this person's blood group we don't know, right. This person has placed 3 drops of blood and mixed antibody A, B and D. This person has mixed now. So, what will be the result after mixing? After some time you can see this is clear. Which is clear? This is in the form of liquid. This is a liquid, but these two have clumps, dots like structure. Can you see? It is not a clear solution. You have small dots in the B and which one, positive or negative also you have dots, which means this is the blood group. Why? Because what we added in the second drop, we added antibody B, no? You have clots in this antibody B because this person's blood has antigen B. Okay. How this concept works is, if a person has antigen A, and when you add antibody A to the blood, it will clot. Okay, it will clot or there is agglutination. Similarly, when a person's blood has antigen B and when you add antibody B to it, there will be agglutination. That has happened here because what we added in the second bottle, antibody B we added. So, that antibody B has clotted with his blood antigens. So, we can declare that the blood group is B blood group. Okay. Now, when I have added when I have added antibody D to his blood, again there is a clot. I told you whenever there is a clot, it means presence of. So, only if this person's blood has antigen D, when I add antibody D, it will clot, no. So, it is evident that this person has antigen D, which means he is positive. So, what will be the result of his blood group? B positive, my blood group. Okay. So, I have explained how to test the blood group. Okay, what about the other blood groups? How it will appear for other blood groups also? Quickly, I will give you a presentation. Okay. What about A positive blood group? So, this follow the same rules. The first drop of blood should be added with antibody. So, everything is antibodies. Okay. So, first we'll have antibody A, antibody B, antibody D. Okay. So, here there is clotting or agglutination, which means only if the person's blood has antigen A, this when I add antibody A, it will clot. So, this person, see here you can see clotting, but this is a clean fluid, which means there is no antigen B here. Why? Because I have added antibody B here, but if there is antigen B, it will clot. No, there is no antigen B, so no clotting. So, now I have found that his blood group is A blood group, and here in the D also there is agglutination. So, he has antigen D, that is the rhesus monkey's antigens. So, he can be declared as a positive person. So, A plus or A positive is the blood group. In this case, A negative, again there will be agglutination in A. In B there should be no agglutination, and D also no agglutination. So, it is absence of D antigen. So, this person's blood group is A negative. Clear? Then about other blood groups. How it will appear for B positive and B negative? B positive person, A, there will be no agglutination because this person will be having B antigens. So, when I add B antibodies, in that place there will be agglutination, and in D also he will have agglutination if he is positive. So, B positive. What about B negative? In B negative also, A antigens he will not have, B antigens he will have which will react with B antibodies, and there will be no D antigens, so here it is negative, so B negative blood group. What about AB positive? AB positive person, when I add antibody A and antibody B, both will show agglutination because this person will be having both antigen A in his blood and also antigen B in his blood. So if I add A antibody also, that will go in clump, and if I add B antibody also, it will go in clump. So for AB blood group people, both in A and B there will be agglutination. Here if he is positive, D also will show agglutination, and if he is negative for D, his blood group can be declared as AB negative. Do you understand? If you have further doubts, you can ask in the next Zoom session. Then what about O blood group people? See, in the first slide I have showed you a chart, no? So there I have told you, do you remember the picture? The red blood cell had no antigens at all, just lines were present, no? They do not have either antigen A or antigen B. So when I add antibody A and antibody B, what will happen? There is no agglutination because there is no antigen at all for the antibody to go and lock to it. Since O blood group person do not have either antigen A or antigen B, there is nothing to get clotted or agglutinated with when we add antibody A or antibody B. So the blood remains a fluid, and for positive people, then the presence of D antigens will lead to clotting when you add D antibodies. What about O negative people? All the three will not be agglutinated. Blood will remain a liquid. The absence of D antigens will give a liquid nature for the blood. Due to the absence of antigens, when you add antibodies, it will remain a liquid. This is about O negative people. Hope you will be clear with the blood group testing all now. If you have further doubts, you can contact me personally, or you can clarify in the next Zoom session. That is all for today's biology class. Until I meet you in one more biology class, you all take care and bye-bye.